So we basically just got into the very beginning of chapter 11 on Wednesday, and it's talking about antibiotics, antimicrobial drugs, right? We talked about the forms of containing and killing microbes using disinfectants and antiseptics and germicides and that kind of thing. This chapter gets into the actual drugs that go in and help take out microbes when they start an infection, after they've started an infection, or before they've started an infection. All right, there we go. So when we talk about antibiotics, one thing is that, you've got to get some of the terminology out of the way. Antimicrobial chemotherapy. That is just giving drugs that hopefully destroys the microbe, but doesn't harm the host. And of course, cancer chemotherapies aren't as selective as these antimicrobial chemotherapies, because cancer chemotherapies often can't really distinguish between the cell, a cancerous cell, and a non-cancerous cell. But with the antimicrobial chemotherapies, they are targeted for, mostly, if it's bacteria, prokaryotes, right? And because us as the host, we as the hosts are eukaryotes, that allows us to come up with drugs that can decipher the difference, right? Essentially. These drugs are going to be largely produced naturally. They're going to come originally from other microbes that have used those drugs to kill off other microbes that try to invade their space. And then from there, we can either make them synthetically, or we can make them like semi-synthetically. But there are some drugs that have just been, it's been a rational design of a molecule that we know will target, or that we presume will target a microbe. But most of them, like I had mentioned on Wednesday, most of them are like drugs in general that come from plants that have a natural origin. And of course, there is no such thing as the perfect drug. We have a list of characteristics that I'll get into, and no drug really fits the bill for all of those. So what we try to do is we try to find something that is a balance with as much characteristics checked off as possible. And so what are these characteristics? Well, again, you want it to be selective. You want it to kill the microbe, but not harm the host. Ideally, it's microbicidal, not microbiostatic, right? Microbicidal means it kills the microbes. Microbiostatic just means it keeps the microbes from expanding their population. We want to kill those things inside of us, right? Not just keep them at bay. We want it to be effective long enough so that it can do its job before our bodies break it down and metabolize it, okay, and excrete it. So the pharmacokinetics of these drugs have to be such that they don't lose their potency before they can be delivered to their target. We also ideally don't want a drug that the microbes are going to become resistant to, and then we're back at square one. It's nice if the drugs can complement our immune system defenses, right? And we have drugs like vaccines and antibodies. There's been some new exciting data with some antibody therapy for COVID-19. So those are going to be examples of things that are used to boost your immune system or to complement your immune system. It has to be strong enough that it doesn't get diluted once it hits the bloodstream or once it gets into that tissue that it needs to go to, that it still has its efficacy. It needs to be able to get to the site of infection, right? So, for instance, with the influenza vaccine, it can be a shot or it can be a nasal spray, just right up straight into the lungs. Okay, you're getting it right there, right away. Ideally, it's reasonably priced, although we know that drugs are very rarely reasonably priced. But that's something to consider, at least. Um, and then also, you don't want this drug to disrupt the host's health by causing allergies, okay, or predisposing the host to other types of infections. So again, there aren't really any drugs that check off all of these characteristics, unfortunately. And honestly, the more characteristics they check off, a lot of times, the less reasonably priced they're going to be. But again, we try to balance this out as much as we can. What's an antibiotic? An antibiotic is going to be something that microbes produce naturally on their own. Okay. And a lot of the antibiotics that we have specifically come from streptomyces and bacillus. So streptomycin, I'm sure you've all heard of that. That comes from the streptomyces bacterium. Mold, we're going to have penicillium mold, which produces penicillin, and the cephalosporium, which produces cephalosporin. Okay, those are antibiotics, anti-life, right? And so again, these were evolved in these animals not for our benefit, but for their benefit, to keep things away from them. Here's a table of some of the specific genera of microbes that produce a lot of our current drugs and the type of microbe they come from. You don't have to memorize this table, but it's there for you to, like, again, as always, for your own information. And then, I do expect you to know examples of certain things. So review this table for that. Here's some terminology that I'm not going to go through each one because we'll talk about them throughout this chapter, but just to highlight a couple: prophylaxis is going to be using a drug before someone's sick. Semi-synthetic drugs are going to be drugs that are modified in a laboratory after being isolated from a natural source. So they take that isolate from a microbe and they tweak it a little bit. Or instead of having the microbes produce it per se, they manufacture it by some other means. Synthetic is going to be entirely laboratory created. Okay, so just designed and produced in the laboratory. De novo. Narrow spectrum, broad spectrum, we've talked about those already. So again, prophylaxis before a person is at risk. So vaccines are a good example of a prophylaxis. Chemotherapy is just going to be the use of drugs to treat a disease. In this case, we're talking about microbial chemotherapies and antibiotic against life. Now, these are the things I want you guys to focus on, because the rest of the chapter is going to be devoted to these mechanisms of drug action. And then we'll have examples and what they do specifically. But the 5 major components that you need to know are these. 1, inhibiting cell wall synthesis. So again, once the cell wall is built, it doesn't just stay a cell wall and it's stagnant, right? It constantly needs nutrition so that it can put new lipids and new biomolecules to maintain that cell wall. So if you can no longer utilize nutrients and deliver them to the cell wall, then your cell wall will eventually break down. There are other components that break down the cell wall specifically. So instead of preventing the maintenance, it directly attacks the cell wall. The cell wall is that protective barrier, just like our skin. And once that's compromised, it loses integrity and it falls apart. Interfering with DNA or RNA, so preventing the microbe from reproducing, or preventing the microbe from making proteins that it needs to assemble itself or just to maintain itself. Inhibition in protein synthesis, right? Blockage of key metabolic pathways. So those are the 5, and that's what we're going to talk about in more depth now. Here's a little schematic showing that. So this is just a generic diagram of a bacterial cell. And so for one cell wall inhibitors, it's going to be attacking this outside, this cell wall, and blocks synthesis and repair. And then it gives you a list of different types of antibiotics that do that. And it should be pretty familiar. I mean, I think most of you have heard of penicillin, cephalosporins, vancomycin, that kind of thing. Cell membrane, that's going to go to the cell membrane right here, beyond the cell wall. And that's going to lead to the breakdown. And then polymyxins are the most common version of an antibiotic that attacks the cell membrane. And then quinolones are an example of DNA, RNA inhibition, so on and so forth. So protein synthesis inhibition can be done usually by targeting the ribosome. So you block the ribosome in some way, or you alter the substrate of the ribosome so that it can no longer take that RNA and translate it into the necessary proteins needed. And then again, here are a list of different categories of antibiotics that attack either the 50 or the 30 or both subunits of the bacterial ribosome. And then finally, metabolic pathway inhibitors. Okay. So that's an overview of the 5 different targets. Again, here's a table. You don't need to memorize these 2 tables, but it gives you a really good breakdown of, for bacteria, for instance, what are the different drugs that are used to treat these bacteria. And again, it depends on the type of bacteria. These are not all just broad spectrum that kill off any type of bacteria. Sometimes it's specific to gram-positive or gram-negative. But it gives you a list. So penicillin, for instance, that comes from the mold, and that targets certain bacteria, and it does it by cell wall damage. Okay, so that's an example. Cephalosporin, same thing. And then you can get into these different ones like fluoroquinolones. I don't know if you've heard of those, but they block DNA replication and RNA synthesis. Tetracycline, you perhaps have heard of that. That inhibits protein synthesis. So it gives you a nice breakdown, like I said, of all the different drugs, what they target, and how they target microbes. That was bacteria. Here's fungi. So amphotericin B is really the most common one. And it, again, tells you that how does amphotericin B attack fungi? Well, it makes the, it causes a loss of membrane permeability. Okay, so the membrane needs to be semi-permeable, right? And so it can't do that. It can't excrete waste. It can't take in nutrients, and the cell eventually dies. Quinines, again, buildup of toxic waste in the parasite cells because of preventing that permeability, or those factors that carry the waste to the different pumps that excrete it. And then viruses. And we'll talk about we'll talk about as much as we can today, these things in more specifics, but not everything. So these tables are here for you guys to just examine, but not necessarily memorize each and every one. Any questions so far before we get into the 5 more specifically? Okay. So the first type, antimicrobial drugs that affect the bacterial cell wall. Again, penicillins, cephalosporins. What do? They block the synthesis of peptidoglycan. Remember peptidoglycan, right? It's the cross-linking NAGs and NAMs that make up that. And it's going to be thick. The peptidoglycan is going to be thick in gram positive, thin in gram negative. And because it attacks the peptidoglycan, and because gram negative bacteria have that outer membrane that kind of cover the peptidoglycan, these are not going to be as effective on gram negative bacteria, okay, because they can't get through that outer membrane, that candy coating on the outside. And so these class of drugs are going to target more specifically gram positive, okay? But they are still broad spectrum to a certain degree, because some of them are able to get beyond that outer membrane and into the gram negative bacteria. Drugs that disrupt cell membrane, essentially maintenance, right? So it's going to prevent metabolism, and therefore the metabolism is out of place. It can't get its metabolites sent to the cell membrane to maintain it. Polymyxins are good examples of this. They interact with the phospholipids, because remember, cell membranes are made up of phospholipids, just like ours are, eukaryotic cell membranes. That's one thing they have in common. And so what happens is you get this kind of leakage, okay? So they can't, now they're no longer selectively permeable, they are just permeable, and they'll break down. And this is a good one to use on gram negative, because these attack that outer membrane, okay? Amphotericin B and nystatin, those act with fungi, remember, and they do that by forming these complexes with the sterols on the fungal membranes. Remember, we talked about sterols a little bit. We said that they're alcohol molecules that are made up in cholesterol. There are some bacteria that don't have that coccus or bacillus shape. They have a more amorphous shape because their membranes are made up of sterols. These fungi have sterols because fungi are eukaryotic. And so they just kind of mess up the sterol array. And so again, making it a leaky membrane. And here's just an example of that. So the polymyxin B, it's going to intercalate into this cell membrane and eventually break it down. And now you have these big gaping wounds, if you will, in the membrane, and that can't support life. Things get in, things get out, and it's effectively killed. All right. So the first two really target the actual microbe itself, right? As well as the last one that gets in the way of metabolism. But the ones that involve disrupting DNA and RNA synthesis, those are going to be more like, well, we're just going to prevent you from growing more, your population. And eventually you will die. So it's still microbicidal, because the cell, not only can the cell not divide, but if it can't upregulate certain genes, or if it can't take those upregulated genes and transcribe that into RNA and then make proteins, that cell will also die, right? So it's a kind of a 1-2 thing. It prevents the cell from maintaining itself by making necessary proteins. It also prevents it from undergoing cellular division to make babies. Okay, so that's what we're talking about. Blocking the synthesis of nucleotides or inhibiting replication. So you can block the synthesis of nucleotides by having these things called analogs, which look like the nucleotides from a molecular perspective, but they're slightly different. So when they can be used to make that new strand of RNA, but once you go to translate it, you can't translate it. Okay. Sometimes it can also pop off the polymerase. So it intercalates into the DNA, and once the polymerase is chugging along that DNA strand, it hits that analog and just pops it off. Okay. Chloroquine. You guys all know chloroquine, right? Touted as a anti-COVID drug, which has been shown to have really no efficacy in treating COVID. But nonetheless, what it does is it binds and cross-links the double helix of DNA, okay? And so what happens is that DNA helicase, which, anybody know what DNA helicase is?
I'm guessing it's an enzyme that helps DNA form a helix, something like that.
Doesn't it unzip it?
Yeah, so you're on the right track. It kind of does the opposite, but yeah, helicase, so anything that ends in ace is going to be an enzyme. But we have these things called topoisomerases and DNA helicases that help unwind, because remember, the DNA is all super coiled into these chromosomes. It helps unwind the helix so that the polymerase can just target one strand and replicate. Okay, so you have a topoisomerase that then begins to unwind the chromosome, which, let's think of like a ball of yarn, right? So that's the chromosome. The topoisomerase unravels the ball of yarn. And then, if you guys are familiar with yarn, it's not just one strand. Usually it's several strands that are kind of woven together. The helicase is now going to take those strands that are woven together and unzip them, so that now you have something to transcribe into new RNA. And so by cross-linking that DNA, it can't be unzipped. So it's just effectively, a statue, if you will. Drugs that block protein synthesis. So luckily, eukaryotic ribosomes have a different size and structure than prokaryotic ribosomes. And so we have specific antibiotics that can selectively attack the prokaryotic ribosomes. It's not that they look and see that this is this and that is that, right? They don't have eyes. It's just they have a selective design in their structure that only binds to a specific type of ribosome. So would this be something that's, what kind of microbes would these drugs be used on? What's an example?
Bacteria.
Bacteria, right? Not a good fungal drug, right? Why wouldn't this be good on fungi? Because fungi are eukaryotic. Yeah, exactly. Then we have things like aminoglycosides, streptomycin, remember from streptomyces, and gentamicin. They're going to insert on the 30S subunit, so that smaller subunit of the ribosome. And as a result, that's going to cause misreading of the messenger RNA. Okay. So it's going to not make the correct protein. Tetracyclines, I'm sure you've all heard of tetracyclines. Those block the attachment of the transfer RNA on the A acceptor site, so the adenosine acceptor site, and stop further synthesis. So remember, you get a messenger RNA that's transcribed from DNA. It leaves the nucleus, right? And then a transfer RNA does what? What does a transfer RNA bring?
Don't they bring the amino acids?
Yes, they bring the amino acids to the ribosome, the proper amino acid that corresponds to the codon, the 3 different nucleotides. Okay. And so what this is doing is it's preventing a specific transfer RNA for the A nucleotide so that now we can't put our A's where we need to. Okay. And so here are some examples, right? So the aminoglycosides, they are going to bind again to this 30S subunit. And you guys remember what the S stands for?
Size.
What?
Size.
Spedbergs. Yes, it's the size. It's particularly the rate of sedimentation. So how far that particular molecule migrates through a gradient, usually a sucrose gradient. But anyway, they bind here, and now as a result, they interfere with the translation of the codons in this mRNA strand. Chloramphenicol prevents the formation of the peptide bond, okay? So what's a peptide bond? It's a bond between 2 amino acids. So now, think of protein as like a long train, and each car on the train is an amino acid. If you have a break where 2 cars in the train aren't connected, you're not going to have the whole train. If you don't have the whole train, you don't have the whole protein, right? And now it can't do its function. There's a process where you prevent the initiation of the 2 subunits coming together. Okay, so let's see, oxazolidinones, those are going to bind to this amino acid. And then when it tries to latch down onto the 30S subunit, that docking port is now blocked. Tetracycline, that's going to block these tRNAs, and so they can't bind to the site. They can't deliver the adenosine, particularly. And then erythromycin, what does that do? That binds to the actual site of the messenger RNA. And so now you can't undergo translocation, which is just saying it can't that ribosome can't process, it can't travel along that messenger RNA and read it and then translate it. Okay, so these are all different, like, so ribosome is implicated in all of these antibiotics, but in different ways. Okay, and be familiar with some of the ways in which the ribosome is blocked. Okay, several here. Drugs that affect metabolic pathways. So think competitive inhibition, meaning they will bind to a site that is supposed to be dedicated to some other enzyme or substrate. And when you block that site, now you can no longer carry out that metabolic process. It's kind of like how caffeine works. Caffeine mimics a certain molecule that binds to certain receptors in your brain. And when you wake up, you flushed out all of those receptors and they're waiting to be bound to something. The caffeine comes in and it binds to that receptor, but it's different enough from the molecule that natively binds it that it doesn't activate the receptor. It just blocks it. Okay. And these receptors over time, as they bind to their native molecule, that sends the signal that you're getting tired. Okay, so caffeine kind of interferes with that process, and that's why it keeps you more alert. Now, the body also responds, and it just makes more receptors. And so after a while, you get caffeine tolerance. But this is a similar kind of process, right? You bind to something, and now the native molecule that was intended to get there can't. Same thing with antibodies, right? As an antibody therapy, you're binding to something and blocking it, or in the case of microbes, designating it for destruction. A synergistic effect, that means that we have a combination of different inhibitions that lead to an overall breakdown in the metabolic pathway. Okay, so synergistic, it's the sum of these two or more antibiotics coming together to form a greater effect. Okay, so oftentimes you'll have a combination therapy of antimicrobial drugs so that they act on different things in combination. Okay? Any questions on this so far? No? Pretty straightforward. Okay. So let's start looking at the different drug categories. Okay, there's a lot. There's over 280 different drugs, and we've classified them into about 24 different families based on their function, their mode of action. And here's the 5 major ways of targeting microbes that you need to be familiar with. And then here are some of the drugs that we've mentioned. So for the first one, beta-lactams. These are broad spectrum, and they're very commonly used to attack all different types of bacterial infections, and penicillin and cephalosporins. Those are the most common ones. And it's a beta-lactam because it has this 3-carbon 1-nitrogen ring right here. So this is a, can you guys, I don't know if you can see my little cursor or not. So here's a carbon, 1, 2, 3, and then this blue is a nitrogen. Okay. And what that does is, again, that interferes, that molecule inserts into the cell wall and then prevents maintenance. And it prevents the incorporation of new components of the cell wall into it. Okay. And like I said, this accounts for a large majority of bacterial antibiotic drugs. However, there are non-beta-lactam drugs as well in this first category of cell wall inhibition. And so a drug like vancomycin is going to be narrow spectrum, but it can be used in infections that are penicillin or methicillin resistant, or if the patient is allergic to penicillin. Okay, this can be used as a substitute. because it's toxic to a certain degree, and because it's hard to administer, and because it's narrow spectrum, it's not as widely used, but it can be used as a backup. Bacitracin, also narrow spectrum, produced by Bacillus subtilis. And that's usually what's going to be a component of things like Neosporin and stuff like that. Any questions on this? Okay. Second category, again, I've mentioned this, but polymyxins, narrow spectrum. They are going to be used for things like Pseudomonas aeruginosa. That has a very high tolerance to a lot of different drugs. And then in the case of really bad UTIs, urinary tract infections, they can be used as a kind of as a last resort to treatment. Fluoroquinolones, they bind to DNA gyrase and topoisomerase. I mentioned topoisomerase earlier, right? So they prevent the DNA from unwinding to access that particular region of the DNA that needs to be transcribed into RNA. These are going to be broad spectrum. However, there is concern that they're being overused, and overuse leads to resistance. And so again, it's being actively monitored right now. By the time you guys, those of you going into nursing, by the time you finish, it'll be interesting to see where this is at. Aminoglycosides, a lot of them come from actinomycetes, which perhaps you might remember as one of those categories of bacteria that are found in the gut, and they get there because they're a common soil bacteria. And some are bad and some are fine. And we eat a lot of dirt, not on purpose, sometimes on purpose, I guess, for some people, but you're gonna get your gut colonized just by being around soil. Relatively broad spectrum, they inhibit protein synthesis by binding to a ribosomal subunit. Okay, and so now those subunits can't come together. Particularly useful for aerobic gram-negative rods or bacillus, right? And some gram-positive bacteria. And here are some examples of those. Okay. So what am I expecting you guys to know for quizzes and finals and stuff? I expect you to know these 5 different types. I expect you to know examples of broad spectrum versus narrow spectrum. And I expect you to know some examples of drugs for each, okay? Just like the DNA viruses and the RNA viruses. I don't expect you to know all of them. I want you to give me examples, though, for each category. Tetracycline, broad spectrum, blocks protein synthesis. How? By binding to ribosomes. So that would be your example. If you get to number 4, drugs that interfere with protein synthesis, then one option you could do was tetracycline. You'd write tetracycline, broad spectrum, blocks protein synthesis by binding ribosomes, right? Now, if you knew some examples also of what it treated, that would probably be good, right? Icing on the cake. If you knew that tetracycline is largely used for sexually transmitted diseases or Rocky Mountain spotted fever or Lyme disease, then that would be also good to know. Chloramphenicol, also broad spectrum, blocks peptide bond formation. Okay, so this is the one, it doesn't prevent the ribosomal subunits from coming together. It prevents the amino acids from coming together. So you can't form that protein train. This is an example of an entirely synthetic antimicrobial. Okay, it can be very toxic to the host, so it doesn't check off that selectively aggressive category. And so it's used with caution and discretion. Oops. There we go. Macrolides, erythromycin, right? We've all heard of erythromycin. It has this lactone ring right here and these sugars attached to it. And what that does is it attaches to the 50S subunit, not the 30S. Broad spectrum, again, broad spectrum. Fairly low toxicity. That's why it's used commonly, right? That's why we've all probably had erythromycin at some point in our lives. Prophylactic, prior to surgery. So this would be an example of a drug that's used prophylactically. Also good for microbes that are resistant to penicillin, like gonorrhea and syphilis. And then there are some newer semi-synthetic versions. Semi-synthetic meaning they came from a natural source, a natural microbe, and then have been tweaked in the lab. Okay, now finally, the fifth category, drugs that interfere with metabolism. These include sulfa drugs, narrow spectrum though, okay? Some things they can do is block the synthesis of folic acid. Bacteria needs to make folic acid. And so if it doesn't get that metabolite, it won't survive. This can be used on things like Shigella, UTIs, and also protozoan infections, so eukaryotic organisms. Can be used on burns and eye infections because it's not as caustic as other topicals. Certain topicals can be applied to the skin if the skin is not compromised somehow. And so if you have burns, which I'd mentioned on Wednesday, which is a great way to get infection because you've compromised that outer protective layer, you don't want to put something on that burn that is going to exacerbate the pain that the patient's already experiencing. So some of these sulfa drugs are good options for that. And of course, your eyes are highly sensitive, right? So you don't want to put something in your eye that's going to burn it or be uncomfortable. Trimethoprim is going to be one of those synergistic drugs used with a sulfa drug for the treatment of UTIs. Okay, so there you go. There's an example of a synergistic treatment. These are some new classes of antibiotics, but I'm not going to get into these. I'm not going to test you on these. I just put this in here because, again, for those of you going into nursing school, you'll probably have to get a firmer grasp of the newer classes that are coming out. And so this is for your own information so that it's a little bit familiar in case you come across this again. But highlighting that, these are being developed because of antibiotic resistance, right? So for instance, this one at the very bottom, Zyvox is used to treat MRSA, okay? Because what else are we going to use, right? Not a whole lot of options. Any questions? Those are the 5 different categories and specifics related to them. Any questions or comments before we chug along? Okay. Antifungals. Like we said, fungus is eukaryotic. So if you start perturbing the natural processes of these eukaryotic cells, you could very easily start perturbing the eukaryotic processes of your own cells. So it makes a little bit more challenging. Okay. That's why most of our chemotherapeutic drugs are designed for bacteria. And that's why they can't also work on fungi. Because a drug that will kill off fungi could also potentially harm the patient. And so we have 5 different groups that are developed specifically for fungal infections. And here are those 5. For instance, the macrolides, they bind to the fungal membranes and cause loss of selective permeability. I mentioned that earlier when I said amphotericin B, okay? It binds to these fungal cells and it makes them no longer be able to import the things they need and export the things they don't want. And so they eventually become toxic and die. The griseofulvin is for stubborn cases of dermatophytes. Where do you think dermatophytes live?
Under the skin.
Yeah. Synthetic azoles, broad spectrum. Flucitocin is an analog of cytosine. So remember I gave you the example of certain analogs that will either intercalate into the DNA or be confused as an actual nucleic acid. This one is an example that mimics cytosine. And then when the polymerase comes along, it encounters that that analog. It's like, I don't know what this is. The nucleotides next to it were tricked. It's like the Trojan horse, right? They think it's a cytosine, but the polymerase doesn't, and then doesn't know what to do, and maybe it puts in a mutation, or more likely it kicks off the polymerase. And then capsifungin that damages the cell wall. So there are some things that can be used, but again, it's a lot more challenging because of this eukaryotic nature of fungi. Antiparasitic chemotherapy. So usually when we think of parasites, in this case, we're talking about protozoans and worms. So chloroquine, quinine, these were anti-malarial drugs. And then we have certain sulfonamides, tetracyclines that can also be used on different protozoa. Anti-worm drugs or anti-helminthic drugs are going to be things like mebendazole, broad spectrum. What that does is that interferes with the microtubules. And remember, microtubules are those things in the cell that are like the highway, kind of like the actin filaments. The microtubules allow translocation or movement of certain proteins or certain molecules from one part of the cell to another part of the cell. Niclosamide destroys their scolex, and we haven't talked about worms yet. That's later on, but the scolex is like this hook, these hooks on the outside of the mouth that allow it to attach to your GI tract. Okay, and so if that scolex is destroyed, it can't latch on. It's analogous to the fimbriae of bacteria, right? Adhesion. Antivirals. Remember, viruses are considered parasitic as well. All different types of ways of interfering with viral infection, right? You can block entering into the cell. If a virus can't get into a cell, it can't replicate. It has to be inside the cell. obligate intracellular parasite. Another mode is, well, we'll let the virus in, but we're not going to let it replicate. Okay, we're going to cut out translation. We're going to cut out transcription. We're going to have, again, these nucleotide analogs that mimic regular nucleotides and then sabotage the translation or the transcription process. And here are some examples of those and the different viruses that they target. And then another category is preventing the maturation of the viral particles. So remember, a virus, going back to virology, we talked about how viruses have to get in the cell, they have to uncoat, and then they have to replicate, and then they have to go back to a specific site in the cell and reassemble, and then leave the cell by budding or by lysing the cell. And so we can prevent that from happening. We can say, okay, the virus gets in, the virus uncoats, the virus replicates, but we're not going to let the virus mature to new virions or viral particles. Okay, so some of those are protease inhibitors. What do you think a protease inhibitor is? Anybody? What's a protease?
Protein enzyme?
Yeah. But all enzymes are proteins, so specifically it's an enzyme that cleaves proteins, or it cuts them. And a lot of viruses that have just one strand of, say, RNA, for instance, they need to cut the proteins into different segments, and then those different, peptide cleavages become functional. Okay, so what I'm trying to say is, rather than have an RNA segment for every protein that is needed, then you'd have to package a bunch of different RNA segments into this little viral package. You just have one long strand of RNA, and you translate one long polypeptide, and then you break it up into the capsid protein. Another part gets broken off, and that becomes the RNA-dependent RNA polymerase, right? Another one gets broken off, and maybe that's part of the tegument or the matrix protein. So if you have an inhibitor of the protease, now all you have is this long polyprotein, and it can't be spliced or diced into its different structural proteins. Okay. So these are some ways of preventing viral infection. And here are some specific ways, specific examples of drugs. So Fusion, that's anti-HIV. It prevents the binding of the virus, so it can't fuse to the cell and enter. And then Maravirac covers up the receptors on the cell, so it can't even adhere to the cell, right? Remember, a virus needs a receptor to gain access inside the host cell. And that's what gives viruses their specificity. That's why certain viruses only infect certain types of tissues, because certain types of tissues have specific receptors, okay, indigenous to them. So with the new coronavirus, that binds to the ACE2 receptor. So again, any cell that has that ACE2 receptor is fair game for COVID. And once you, but you can't just necessarily block any receptor, because if that has a specific physiological function, then you're in trouble, right? So we have to pick the right mechanism to inhibit that will prevent viral infection, but not, maliciously tweak needed processes of the host. Tamiflu is for influenza, And so that's going to, it's not going to stop the early cycle, so entry in uncoating and replication. It's going to stop it toward the end, toward the maturation process. Okay. And so here's a little diagram of this. So right here, we're taking this drug and we're putting it on the cell membrane so that once the virus binds, it can't enter. Okay, it can still bind, but it's just stuck on the outside. Doors are locked, no entry. Okay. Second version, we have this drug that binds to the specific cell that the virus needs to get in. So it just bounces off. Can't find a place to get in. Right? It's like you got a big litter of puppies, and the mama dog only has a certain number of nipples, right? And so that runt of the litter, try as best as it can, it just can't get entry. I don't know. I just came up with that off the top of my head. I don't know if that's a good analogy or not, but anytime you mention puppies, you can't go wrong, right? And then Tamiflu down here, it's going to prevent. So the flu virus gets in, it docks, it gets in, it unpackages. Now, interestingly, the flu. What kind of virus is a flu virus? DNA or RNA?
RNA.
RNA. This is one of the few viruses that's an RNA virus that goes into the nucleus. Because remember, we said most DNA viruses, they have to go into the nucleus to then get RNA made. But most RNA viruses don't have to enter the nucleus. They've got their RNA, they just replicate in the cytoplasm. So the influenza virus is an example of one that is in the nucleus. And then, so it has all 7 segments that go into the nucleus, come out of the nucleus, we get the different products, but then now that neuraminidase, right? Hemagglutinin, neuraminidase, H and N, those spike proteins, they cannot assemble. And so the virus is stuck inside the cell. Okay. Acyclovir prevents herpes, and it does so by mimicking the nucleotides, right? So it's an analog that is convincingly similar to nucleotides, and so they're incorporated. But then, again, the polymerase cannot deal with them. These can be oral medications, they can be topical treatments, just depending on the nature of the herpes. So if it's an oral or genital herpes, or if it's chickenpox or shingles, it's going to be different ways of delivering the medicine. Ones that get in the way of nucleic acid synthesis. So acyclovir, nucleoside analogs, and reverse transcriptase inhibitors. Remember what reverse transcriptase is? It's used for negative sense RNA viruses that need to then transcribe that positive sense segment that then can be translated into protein. So again, you're inhibiting, like you're taking different strategies depending on the nature of the virus. Is a reverse transcriptase inhibitor going to be effective at treating a virus that doesn't need reverse transcriptase? No, right? So all these microbes, whether they're bacteria or fungi or worms or viruses, are all going to have a multitude of different drugs that are specifically designed for a very particular type of microbial infection. And you can't use the same drugs on a bacterial infection that you can on a viral infection. And within certain bacterial infections, whether it's gram or negative, gram positive or gram negative, you cannot use the same drugs. And this is the same problem with treating cancer. Okay. People talk all the time about a cure for cancer, a cure for cancer. Well, that cancer is a lot like microbial infections. There's not just one type of cancer. There's not just one type of mutation that drives a particular cancer, right? You can have breast cancer, and that can be HER2 positive, HER2 negative. It can be a bunch of different drivers all acting at once. So similarly, we need to customize drugs to take on the specific nature of the disease. Otherwise, you're just wasting your time. Okay. And then here's a diagram for these and shows what they do. So acyclovir, it enters the cell, it goes into the nucleus, and then it binds to the polymerase. All right. So you can't get replication of the genome of this virus. In this case, the epivir is going in and it's binding to the so you have reverse transcriptase here, and it's binding to that, and so you don't get any reverse transcription into the positive sense strand. And then interferons. Do you guys remember interferons? What's interferon? We're almost done, I think. What's interferon, guys?
So it release the toxins to the release the signal to the neighboring cell when it dies.
Yeah, so it is a signaling molecule. One of the things that it does is it's released as a warning system to neighboring cells to say, hey, there's an infection going on. That can be used as a treatment. And so that would be an example of a therapeutic that complements the host's immune defenses, okay, that we talked about at the beginning. And then this talks about, some of the things that it's used for, and the examples of viruses and the symptoms that they treat. Okay. Last 10 minutes or so, I want to talk about antibiotic resistance, okay? And this is a problem, right? Because people think, well, I'm feeling better now, I'm going to stop taking this pill. And they don't have Batman here to slap them upside the head and say, no, do the whole regimen. Because once you stop, you haven't killed off all the bacteria, you've just killed off enough not to feel bad. And therefore, the ones that are still not dead are the ones that are most hardy, right? And so that is how we develop this resistance, by overprescribing and by undercomplying. I also mentioned that it's natural selection, right? That this happens. You have a large population of microbes. If you just have one or two bacterial cells that are resistant to a particular drug, then everything else dies off. And what's left are these resistant cells. And they are the ones that then take over, right? Just like natural selection. If you have some characteristic that allows you to be more adapted to your environment, then you are more likely to succeed and survive. And therefore, you will then reproduce, and those progeny at least some of them will have that adaptive trait. Okay. And that's how bacteria becomes resistant, through the process of natural selection. And when we don't take our antibiotics to the full course, we facilitate that even further. Okay. Drug resistance. So there's several different ways that bacteria specifically acquire resistance. And we'll go into those different ways. It's either allowing them to tolerate the drug by going, like, it depends again on what the mechanism of action that drug takes on. And if it's attacking a certain fundamental biological process, and this new resistant bacteria has found a way to circumvent that process, then that's a way to get around it. And there's also ways where they've developed pumps, or they overexpress certain pumps that once the drug goes into the bacterial cell, it says, spits it back out. Okay. It could be something that is intrinsic or acquired, meaning intrinsic, it just through the process of random mutations, it generates some mutation that lets it circumvent these drugs. Or one bacteria cell comes into proximity to another bacterial cell, takes its little pilus there and squirts in a plasmid that has that resistant gene in it. And so that would be acquired resistance. Like, here, share the wealth, man. Right? And so this is what we're really dealing with. And, honestly, people are rightfully concerned about global climate change and things like now pandemics. And, will we have fresh water indefinitely? There are lots of concerns. Drug resistance is should be a major concern. Okay. Because once we lose the ability to control these bugs, then where do we go from there, right? So, spontaneous mutations in critical areas, natural selection. That's how it starts, right? Then you have these new mutations, and they can be passed on to other microbial cells that don't have that, okay? Through the conjugation method, right? Passing plasmids using the pili from one cell to another. And we call these R factors, okay? These plasmids that have resistant factors. So, you're not just dealing with natural selection. Now you're dealing with natural selection and then passing on that to neighboring cells. Okay, how does it work? For instance, there could be the inactivation of the drug. Okay, so there can be certain enzymes like penicillinase, right? ACE enzyme. So the bacteria now all of a sudden can produce this enzyme that cleaves part of the penicillin drug. And that the whole thing is important, right? So here we have this bond here between a carbon. That's messy. We have a bond here between this nitrogen and this carbon. And the penicillinase cleaves that bond. And so now you have this carbon that has lost its bond with the nitrogen and is now, because it needs something else to bind to, it binds to an oxygen free radical, in this case forming an alcohol, and now it can't work in its intended fashion. Okay, so it's inactivated the drug. That's one option. Another way is to decrease permeability. So as I said, you have the drug, it enters that bacterial cell, and then now the receptor that was used to internalize that drug has mutated its shape. And so the drugs can no longer bind to the receptor and then be internalized. Okay, so it's pointless. These are not drugs that are going to act on the cell wall, right? These are going to be the types of drugs that work inside to interfere with metabolism or DNA, RNA replication or protein synthesis. The activation of drug pumps, right? So drug goes in, now you have a pump and it says, nope, get out of here, right? So all different methods, but still conferring the same overall goal of not letting that drug do its job. Changing the binding site, right? So remember we talked about all these drugs that involve inhibition of protein synthesis and how they work. They bind to the subunit in some cases. Well, if you change the binding site, and now instead of having that little triangle shape, you have a little circular shape. Now the drug can't bind it, and the ribosome comes together, and it's on, baby, right? So really creative ways, being anthropomorphic about it. Like it's not like the bacteria saying, "Oh, I need to change my binding site of my ribosome." No, they're not. It just happens randomly, but it's super creative in terms of how nature responds to its environment and how there's always something coming along to adapt no matter what. And remember, bacteria are the ultimate organisms at adaptation. They've been here the longest. They excel at adaptation. Give me a low pH environment, sure, we'll figure it out. Give me a bunch of sulfur, sure. Oh, wow, now we have a bunch of oxygen in the atmosphere, okay. Right? And now you're giving me a bunch of drugs that are supposed to kill me, I'll take on that too. They're ready for the challenge. Altering the metabolic pathway. Okay, so let's say, again, a bacterial cell needs to take something and break it down into a particular metabolite or byproduct that it then uses, whether it's to shore up the cell wall or just any of its overall cellular processes that are required for subsistence. And the drug blocks some critical step. So in a normal native fashion, it would go from A to B to C to D to product, right? Think back to the paper we discussed on Monday, right? That pathway of methanol to formaldehyde, right? And so on. You block step B and now it can't go to C. So eventually certain bacterial cells will find a way to circumvent that. Okay. If I can't get to C anymore, I'll get to C1. And then maybe to D1, or maybe, honestly, what some can do is if we're just blocking B to C, we'll create an alternative route and then go back to D, the original D. All right. So it's a workaround. Wow, look at that. Just 2 minutes over, but we finished. Any questions on that, guys? That was a lot, right? I know. Unfortunately, due to the nature of this class, there's a lot of material that we got to get through. But how was that? Did you guys feel good about it? Feel overwhelmed by it? What are your thoughts?
It's a lot. It's a lot, but it seems like it'll be really useful.
Good. It is a lot. I get that. So take it and dissect it. You know, look at one major approach at a time. I'm going to post a homework assignment so that you can, practice, the information and review it. And it'll be on the final in some way, right? This is particularly important stuff because so many of you are going into nursing. I think that it's good to know, the mechanism of action of drugs, right? And then when we get into bacteria of medical relevance, that's going to be important too, and it's going to be heavy also. But my recommendation is that you just review these slide decks. If you want to watch the video a couple times, it takes time, but practice makes permanent. So that's what you got to do. If you guys are free to go, if any of you, actually, I need to go too, because I need to pick up my kids from the bus stop. So I'm going to go, but unless you have any quick questions, pick up your kits. If you don't have your kits, Ryan, do you have your kit?
I gotta pick up my boy as well, and maybe the kid's priority before the boy. So I'll get it in the next 10 minutes here.
Your scobies with your kit. Make sure you get your SCOBY. Elke, your scobies with your kit. If you guys have any questions about how to make kombucha, let me know. Okay, I've worked through a lot of the squishy parts, so I've got some pretty good success. So
yeah, I have mine already.
All right, make some. Yeah, okay. Well, you need to feed it. So get some tea, put some sugar in the tea, and put it in a jar. And don't put a lid on it. Use a coffee filter or a paper towels or a towel. I like to use coffee filters because they're somewhat decontaminated. If you use a dish towel, that's going to have a lot more microbes than coffee filters on it, just from past use and stuff. So anyway, happy to field any questions about your kombucha brewing process. Okay.
Yeah. I mean, the more sugar we add, the more alcohol is formed, right?
Yeah. So just like with certain wines, the flavor is going to be sweeter if you add more sugar and then you bottle it up sooner. If you let it ferment longer and you put a bunch of sugar in it, it's going to have a higher alcohol content for sure. So there's these hard kombuchas that are made, that are like 7 or 8% alcohol, which is a lot, right? But typically, if you just do the regular process, it's going to be less than 1%. So it just depends on what you want to do. But yeah, we can talk offline on kombucha if you'd like.
Sounds good. Yeah. Okay. Thank you.
All right. Well, I'll see you guys. Have a great weekend. And I will see you Monday virtually on campus.